like to introduce Dr. First, our guest speaker. Dr. First is an internationally known for his expertise in research and treatment of scleroderma and rheumatoid arthritis. He has specialized in studying how medications react in the body to treat rheumatic diseases. He is a member, master, and fellow in the American College of Rheumatology. As of 2015, Dr. First has published more than 330 book chapters and reviews, 366 peer-reviewed research articles, and edited 17 books, including 2 editions of a textbook on scleroderma. He lectures and teaches internationally as well as in the US. Dr. First's area of research includes rheumatoid arthritis, scleroderma, and polymyositis dermatomyositis. Dr. First has been a wonderful asset to the Scleroderma Foundation, to the community of scleroderma, and to patients and families actually nationwide. And we appreciate him, and we will always appreciate everything that he does to not only help the foundation, but to help everybody in the community. So without further ado, please join me in welcoming Dr. Daniel First, who will address Clinical Trials They Never Fail.
So hello, everyone. Thank you for that introduction. My mother would have loved it. So what I'd like to do is I'd like to talk to you a bit about why, no matter what the outcome of a trial is, that it really teaches us something. They never fail. Could I get the first slide up, Manuel? So these are the people with whom I work in industry, and they help fund my research. Next slide. So many of you have participated in clinical trials, and unfortunately in scleroderma, these trials often, quote unquote, fail. It can feel like this was a waste of your time, but really, trials that fail are never truly failures. We always learn, and I want to show you some examples of quote unquote failed trials that aren't failures, and why your efforts and other patients' efforts are always a great help and will move towards more successful treatments. Next slide, please. But before I want to go there, I want to talk a little bit about what is it a clinical trial and what's a successful trial. Most trials ask one clear question, and that's called the primary outcome. And that's what determines whether a trial is a success or a failure. For example, the question might be, when we talk about SARS-CoV-2, can a vaccine prevent COVID-19? But there are also multiple secondary questions. And those questions are always asked, and we try and allow for analysis of them. So for example, for COVID-19, how quickly does a vaccine work? How long is it effective? What about side effects? Those are all secondary outcomes. And that requires a good design, and so that the answer is clear. So you have to have a control, for example, a vaccine placebo in these COVID-19 trials. It has to be large enough to get a clear answer. And that's hard in scleroderma. But here with COVID-19, you get 20 or 30,000 patients per group. And then it has to have a clear outcome. So for COVID-19, it would be statistically prevents COVID-19. Okay, next slide, please. What I'm going to do is I'm going to talk about 3 aspects of quote unquote failed trials. One way that they help, even if they fail, is they help us learn which patients are most likely to respond to treatment. And I'm going to pick 3 examples. Collagen type 1, Orencia or abatacept, and cyclophosphamide. Another way they help is to help us design better trials in the future. And for that, I'm going to talk about Actemra, lenabasum, and D-penicillamine. And finally, I want to give an example of how they help eliminate really ineffective therapies. Next slide. Now, drugs go through a lot of development phases, and many of you know about this, but it's worth just refreshing. First of all, they go through 3 preclinical, where they go through animal studies and test tube studies. And that accounts for about 20% of the cost, in fact. And then it goes through a number of phases. There's phase 1, which is first human use. Does it do any major harm? How does the body handle it? That's called kinetics. Phase 2 are the early efficacy trials, and they ask what doses are likely to be effective, and that's called dose response. And then phase 3 is used when we put the treatment in larger numbers of selected patients. Those are the ones that most of you have been involved in. Next slide. But there are 2 more phases. One is called phase 4, and that is where after the drug is marketed, we put many more patients into trials with many fewer restrictions, much more like real life. And finally, a large real life study called REM. And the cost of that, not even including these last 2 phases, is about $1 billion. So these are very expensive trials, drug development costs. Next slide. So let's talk about some of the failed trials. First, we'll talk about the ones that help us learn which patients are most likely to respond. Next slide. Type 1 bovine collagen, which is a fibrous tissue or scar tissue. Hold on. Was done versus placebo. It was double blind. That means neither the patient nor the doctors knew which treatment they were. It was randomized like a coin toss. And it was a 12-month trial. And it tested oral bovine collagen, called C1, versus placebo. And it allowed patients in of any disease duration. Next slide. Here was the result. And you don't need statistics to see that there was no difference between placebo and bovine collagen. The p-value was 0.1. And remember, to be statistically significant, it has to be less than or equal to 0.05, which means there's less than a 5% chance of having it wrong. But, next slide. When we broke it down into patients who had less than 3 years duration versus patients who had longer disease duration, you can see that all of a sudden, in the patients with longer disease duration, there was a difference. It affected the MRSS, which is the skin score. And the p-value was very nice at 0.006. So even though it was a failed trial, it really wasn't a failed trial, because we learned that for this treatment, it was important to look for patients with later disease. Next slide. We're presently looking for some way to fund the next study. Next slide. What about abatacept? That's Orencia. Next slide. Next slide. What it does is it lands on the receptors on the outside of the cell like that, or, next slide, a different version mops up any extra interleukin 6. Interleukin 6 is a protein which is very pro-inflammatory. So it gets rid of it so that it cannot get onto the cell surface and turn on the cells. And there are a number of them being tested, and you can see some of them listed at the bottom of the slide. Next slide. This study examined placebo versus abatacept, and you can see that it went out for about 12 months. And blood and skin biopsies were chosen. And these were patients with early disease. Next slide. So why pick early disease? Well, it turns out that particularly with diffuse cutaneous disease, which many of you have, about 40% of you, what happens is in the first 4 years, the skin changes very rapidly. And that's the time when, and you can see listed here, many of these internal organ involvements begin to occur. It's a little different than the limited disease, and you see that at the bottom, where the skin is not so quickly involved and the involvement of the organs is different. So why pick early disease? Because more changes quickly. And that way we can tell if something's happening, even in a trial which, in terms of scleroderma, is very short, right? A month, 12 months or more, or less, excuse me. Remember we talked about type 1 collagen? Well, that seemed to work further out in that tail. Next slide. Backward, back a couple. There, thank you. When we did the trial with abatacept, it was in a relatively small number of patients, 88 patients. It was a 1-year trial, and this looks at the skin score. And what you can see, comparing drug to placebo, that both improved, but they didn't differentiate. You see, the p-value is not significant, NS. Next slide. So what did we learn from this failed trial? Well, to do that, I want to explain a little bit about what we're doing nowadays, which is called genetic subsetting. And we try and develop what are called genetic signatures. What you see here is in red, genes that got turned on, and in green, genes that were turned off, and in black, genes that are inactive. And each of those little lines at the top represents a different patient. And across the rows are different genes. And I didn't label all of them because there are hundreds of them. But they come together into groups. And there was one group that's being called by most people an inflammatory signature. And you see the pattern of reds and greens. And then below that, you see one that we call proliferative or fibrotic signature. And you see it's a very different pattern of reds and greens. And finally, there's one that's normal-like, kind of not quite like normals, but quite different than the other two. So we generally now think in terms of 3 genetic signatures: inflammatory, proliferative or fibrotic, or normal-like, although there are other systems that are being looked at as well. Next slide. So what do we look at in this abatacept trial that failed? Well, first of all, about 40% of them had the inflammatory signature, 40% had a normal-like signature, and 20% had the fibro or proliferative signature. Remember, they didn't separate for the skin score, or for that matter, by the forced vital capacity or lung test. But look at this up here. If you look at the the left upper portion, that slide, you can see there are a number of lines up there that seem to be pretty flat, and a number of lines that got smaller. And you can see that the lines that got smaller separated from the flat ones. And those are the ones with the inflammatory or the normal-like signature. The same thing is true on the right upper hand, but less so when we talk about the lungs, which was the left. Sorry, I'm getting it all mixed up. But the point is, in the upper portion, that was flat for the placebo, and several of the signatures changed. So despite the fact that the skin didn't change, which was the primary outcome, when we did signaturing, when we broke the patients into their signatures, we found patients who responded. Next slide. So, although it's failed, we learned that it may work in patients with certain genetic signatures. And we do skin biopsies in all the patients. And by the way, the signature is in the skin, not in the blood, although eventually hope to get it in the blood too. But we continue to look for the genetic signatures that work for which drugs. And you can imagine that we may be able to tailor drugs for specific patients depending on the genetic signature. So a failed trial that did not fail. Next slide. What about cyclophosphamide? That's Cytoxan. You're all familiar with this drug. Next slide. Well, what was done was this was a 1-year trial which compared cyclophosphamide to placebo. And it looked at the primary outcome at the forced vital capacity. Remember, that's the test of the lungs. And what you see here is that the group given cyclophosphamide improved more frequently than the group on placebo. You see where it says +5, +10, +15. That's the percent improvement in that lung test, the FVC. And there were more who improved on cyclophosphamide than on placebo. Next slide. At the same time, there were more who worsened on placebo than on cyclophosphamide. You see the -5, the -10, and the -15. So you can see that if you look at the distribution, so to speak, you can see that more patients improved and fewer got worse on cyclophosphamide, and the opposite for placebo. More worsened and fewer got better. Next slide. And in fact, there was a difference of 2.3% between them, favoring cyclophosphamide. So this wasn't a failed trial. Next slide. But what did we actually find out? When we looked carefully, we found out that if you looked at all the patients' FVC together, the difference between the two groups was 2.3%. But in panel B, if you looked at the patients who had a lot of fibrosis, maximum fibrosis greater than 3, and a higher skin score, greater than 23 out of 51, there was a much greater difference, a 9.8% difference. On the other hand, patients in panel C with not a lot of fibrosis and much less skin involvement, there was no difference at all. Next slide. So it was a successful trial, but we learned something well beyond the expected trial outcome: how to pick patients most likely to respond to cyclophosphamide. Point is that we always learn more than just the expected outcomes. Next slide. Let's talk about another example. Another way in which failed trials help is to help us design better trials in the future. And we're going to talk about Actemra or tocilizumab, lenabasum, and D-penicillamine. Next slide. First, tocilizumab or Actemra. You've seen advertisements about this on the tube all the time, for rheumatoid arthritis particularly, and it is approved for that. Next slide. When we did the trial, and this was a 24-week, that's 6-month trial, and then carried it out to 1 year, you can see that the primary outcome at 24 weeks, you can see that vertical line there, and the difference between the red line and the, I don't know what color line it is. If you look at the spread, that's the vertical lines around it, they overlapped a tremendous amount. And so they didn't differ. And when it was carried out to a year, it looked better, but there was still a lot of overlap, and they still didn't differ. Next slide. But when we looked at the lung, we found something very differently. The primary outcome, the skin score that we just saw, did not separate. It was a failed trial. From the FDA point of view, when we went into the FDA with it, the primary outcome failed, and said they called it a failed trial and they wouldn't approve it. However, let's look at the lung. And this is a little hard. If you look at this, you see the -10 at the bottom. That represents the 10% worsening. That's a clinically important worsening. And the green line was the placebo, and 23% of the patients got worse by that amount, whereas only 10% of the patients on Actemra got worse, and that was statistically different. So in fact, the tocilizumab preserved lung function, even though it didn't meet the primary outcome. Next slide. So it failed because it didn't meet its primary goal, but it's very effective in a second goal, the lung. So it actually succeeded. So hopefully we're developing a better measure based on these kinds of outcomes. By the way, we're trying to persuade the company or other companies looking at the same mechanism to go in again to the FDA to see if eventually we can get it approved. It's a long road, but we're trying. So I want to talk about a new measure that's being developed based on these kinds of findings. That is, when one measure doesn't work, another one seems to. Next slide. The measure we're talking about is the Combined Response Index for Systemic Sclerosis, or CRIS. Next slide. This is a two-step index, and Dinesh Khanna and I worked on it for 8 years. Dinesh was the leader. He got it through his fellowship. Took him forever, but we got it. And here's what we found. First of all, it's a two-step process. If a patient has anything really serious happening, for example, scleroderma renal crisis, or a very significant change in the lung, or a significant change in the heart, or pulmonary hypertension, we call that failed. And what we're looking at is the chance of success. So if any of these happen, the chance of success was defined as 0. Next slide. I think we skipped one. Nope. Okay. No. All right. Go to the next slide. If the patient did not meet one of those exclusion criteria, the severe problem, then we looked at these 5 measures: the skin score, the lung, function, the health assessment questionnaire disability index, how the patient felt all over, and how the physician estimated the patient felt. Next slide. So here's what happened. Next slide. We looked for the major problems. Next slide. Next slide. If a major problem occurred, next slide, and next slide, then the CRIS score was 0 and the patient, quote unquote, failed. Next slide, and next slide. So if they did not have that worsening, we went through the other 5 measures, and after proceeding to step 2, we came up with a definition of the CRIS success rate. Next slide. So now, we looked at this after the fact, and you can see here there are 5 different trials of different drugs, and you can see that the skin score failed in all of them because the p-value wasn't 0.05 or less. Remember, we talked about that. So in all of those, if the primary outcome was the skin, the MRSS, they all failed. But now look at the CRIS. And you can see that for 4 out of those 5, it didn't fail, it actually succeeded. So this measure is much more sensitive to response than is the skin score. Next slide. So this just emphasizes it. Next slide. So you can see the two differences. So let's go on to the next one, lenabasum. Next slide. Now, lenabasum is what's called a CB2 agonist. It comes from the cannabis family, a cannabinoid. And what it does is it separates the very primitive response system in our bodies from the more sophisticated one. The primitive one is called the innate, and it's built to respond to infection. And the other one is called the adaptive, and it is to try and deal with various causes of inflammation. For example, scleroderma. And when that second, when this particular receptor called CB2 is blocked, what happens is there's decreased inflammatory proteins, there's increased death of inflammatory cells, there's increased clearance of bacteria, there's decreased fibrosis, and there's decreased collagen. So it should really be pretty good. That is, few side effects, so not a lot of infection, but at the same time, doing the things we like for scleroderma. Next slide. And when it was applied to an early study of lenabasum, and note, this is a 16-week, very short study, and we used the CRIS as the primary outcome. Remember, the first and most important outcome in a very small number of patients. You see that? 27 in one group and 15 in the other. And we know that normally you need 100, 150 patients. So in this trial, we clearly separated the two. 33% probability in the treatment group versus 1% in the placebo group. And when we looked at the clinically significant number, which is in this case 20, it still separated very well. Very hopeful data. Next slide. However, when we did the larger trial with 150 people, it failed to meet the CRIS primary endpoint. A tremendous disappointment to all of us. We really expected this to work. It was a much larger trial. It was a longer trial, 48 weeks, not 16 weeks, and it allowed different background medications. So why did it fail? What did we learn? One thing we learned was that sometimes we have to be very careful about the number of patients in a trial. Maybe we need more. Second, what's the difference between the 16 and 48-week trial? Was there something that happened during that second portion that resulted in failure? And third, what happened with the background drugs? Was it the background drugs that made the difference? We're still working on that, but one probability is that we have to be very careful about the background drugs we use with this particular drug. Again, next slide. Let's go back one. Despite it being a failed trial, we are learning a great deal, and we hope, and there is already another drug in the same family that's being tested by a different drug company. So that in that next trial, what we're learning about design from this drug may be that which makes the next one successful. Next slide. What about D-penicillamine? Now, D-penicillamine is a drug that was around since dinosaurs roamed the earth. It was used for rheumatoid arthritis, and then for a long time, many people used it for scleroderma. Next slide. But when we did the first well-controlled trial, looking at very low dose D-penicillamine versus standard dose in about 30 patients per group, you can see the change. In fact, both groups improved. both the low and the high or regular dose of D-penicillamine. You see that in the graph, but there was no difference between them. You see the P levels. Next slide. What we found was something that turned out to be true in other trials as well, that somehow patients know when to come into a trial. They tend to come into a trial when they're not doing well. And then the natural history of the disease is, after not doing well, on its own, the body tries to heal and the skin gets better. And what this showed that was in both groups, over time, the skin improved. And you see the amount it improved in all of these. Next slide. Another failed trial, but it didn't fail because we learned that patients who enter trials get better even when the treatment doesn't work. It tells us how very important it is to have well-controlled trials. And the open trials may give us incorrect information. So this failed trial was critical in convincing everyone to do controlled trials. Next slide. Finally, how do they help if they help eliminate truly ineffective therapies? For that, I went to a different disease. Next slide. The drug I'm going to talk about for a moment or two was called anti-CD4. So what does that mean? Next slide. What happens is when the immune system gets activated, it interacts with cells called dendritic cells. And those cells present a protein to a lymphocyte type, most of the time called T cells. And then those T cells are activated and they release CD4. They're called CD4 cells. They turn on a different kind of white blood cell called B lymphocytes, and they turn on fibroblasts, and they turn on different monocytes, and all of those in turn result in inflammation and joint damage. And what's going on when patients with rheumatoid arthritis get so much joint pain and damage to their joints. In fact, in scleroderma, some of that inflammation may be driven by the same mechanism. Now, a trial was done, a phase 2 trial. Next slide. Next slide. And what you see here is the result. These are different doses of the anti-CD4 aimed at decreasing those active T cells. And you can see it doesn't take statistics to know that it just didn't work, no matter the dose or anything. Next slide. Even in this really extreme case, it wasn't a failure because it ruled out a mechanism. So we don't have to spend more money on this mechanism. Let's go somewhere else and find something that works. Next slide. So no trial truly fails because we always learn something that'll help the next drug or the next trial. And what I just showed you was these various examples, and there are many more, that tell us that no matter when a drug trial fails, it doesn't really fail. Next slide. Now, I want to leave you with this. This is a picture of Eggs Benedict served on a hubcap. And that's because there's no place like Chrome for the hollandaise. Thank you for your attention.
You would do that, wouldn't you? Thank you, Dr. First. If people have questions, I see one, and I have one that was sent to me. Can you please type them into the chat box, please, and we'll read them out to Dr. First. When you were talking about the different studies that were considered failed or not failed, and you were talking about some of these drugs, and you put up some of these studies from quite some time ago, including bovine collagen, which was my first scleroderma trial with you guys. Could you elaborate a little bit more about those early trials? If I'm making any sense, the message is semi unclear to me.
So I'm not sure what you're asking, but what happened was, in really a long time ago, all we were doing was giving the drug to a bunch of people and seeing if it made them better. And a lot of people got better. Now we know why they got better, because people come into trial at a time when they're ready to get better. So the initial open trials always looked good, but then when a well-controlled trial was done, they did not look good. Now we know why. And based on those early trials, now our trial design is much better, and we don't have this kind of a problem. I don't know if that answers your question, Tina, but that's what I'm trying to, I think that's what I thought you asked.
Yeah, if it's not, I'm sure they'll send me a message. Thank you. Mike, I had a question when I was looking at what you were talking about, and you went into these recent trials, such as lanabecestat, and you were trying to explain the background therapy. Could you elaborate maybe a little bit more on that? Because I'm trying to understand that on some of these trials, if I'm understanding you correctly, because some of these aren't, they allowed these other drugs where, if I remember back in the day, you didn't always have a choice of some of these background therapies. These more modern studies seem to allow more of that. But do you feel, I hope I'm making sense, that these newer trials that are allowing some of these background therapies are the result of the studies not being as clear of the efficacy or the benefit? Did I make sense with what I said?
I understand what's being asked. If you are doing a trial and there's no background medicine, then there's no interference by that background medicine. And so even if the drug isn't very powerful compared to nothing, it can work pretty well. And for a long time in scleroderma, there was just nothing that was seeming to work. But now we have cyclophosphamide and we have CellCept, and they've been shown to work. And so our trials now, generally speaking, are on background of those drugs, because it's not right for people to have to come off all their medicines.
Right.
They have to come off some of them, but not the best of them. And so now all of our trials, nearly all of them, are on background methotrexate, which has been shown to work for the skin, or CellCept, which has been shown to work for the lung. And then we go ahead and add the other drug or placebo to them. So everybody is being treated, but what we're trying to find out is if it's better with the new drug, okay? So they're allowed on the background and added. And I can't be specific because it's not been published yet, but I can tell you that for lanabecestat, that was one of the issues. That is that the background medications worked well enough, so it was hard to show a difference. I'll give you an example. If a drug works 90% of the time, it has to be a heck of a second drug to show 100% response. And so you have to be careful about the backgrounds. Most of the background drugs work about 60% of the time. If that is so, then adding another drug makes a lot of sense. And that's what we're aiming for now. We want better, not just anything, but we want better. Does that help?
Yes, it's perfect. And so when I kind of think of what you're saying, it's like today with some of these combination therapies that we already know work. you're trying to find more combination therapies that work. So I remember everybody had to wash out back in the 90s of practically everything they were taking, go on a placebo controlled trial where you're either going to be on the drug or a placebo, and then see how many people were responding to treatment. Where today it seems like they're allowing these background therapies, but then it makes it more challenging in the end when you guys are doing your analysis to figure out who's responding to the study drug versus the background therapy. But it's more, I don't know, kinder, I guess, to the person participating in the trial. Would that sound correct?
It's the right. I mean, um, what it means, however, is, and it gets a little technical, but there's something called the effect size. What that is the degree of change you expect compared to the placebo group. And if that effect size is small, to get a statistical difference, you need a lot more patients. So now if we have trials where the background drug is pretty good, you're going to need a lot of patients in order to show something.
I see.
The perfect example is nintedanib. What is the name of that? Nintedanib is been approved for scleroderma for the lung, but it worked because the trial was, in part, because the trial was 570 patients, not the usual 150. The difference in the lung was tiny. It was 1.1%. So the effect size, that difference was very small, made up for by a large number of patients. So we've learned that. Now you've got background therapy that needs to be allowed. So now we're going to have larger trials. It's going to be harder to do, more expensive, slower, but that's, as I say, life in the big city.
Yeah, I appreciate that. It's such a it's so interesting to see how far research has come over the years, and I appreciate you explaining that. Could you maybe touch a little bit on placebo and why that was important and continues to be important in these clinical trials, even with background therapies?
Sure. And my patients know this who have been in trials with me. The mind is a really powerful thing. And the placebo takes that into account. It uses the power of the mind to get better. And we know that works. It works for Raynaud's phenomenon. It works for joint pain. It changes the protein in the body. And so the placebo harnesses the power of the mind. And the drug is given to do better than just the power of the mind. In fact, the placebo improves people about 30%. Power of the mind alone. My patients know this. I'm a great placebo responder. I really am. If someone were to give me a placebo for my headache and say, this is a placebo, my headache will get better. So placebos are important. They're not minor. They're an important part of what we do. That's why sometimes using supplements may work or not, but it makes us feel better because we harness the power of our minds. But to get a drug approved, it has to be statistically better than the power of our mind.
Thank you. So when you were talking about some of the analysis and not having a failed trial, could you touch a little bit on the understanding of the data, the analysis behind the scenes that allowed you to find things so how do I say this? You've got the drug that the person's taking, and you might not have found that it was as effective as you would have liked, but sometimes the scientific research that you're doing has brought you into other research or other platforms or understanding something in a gene or a cell because you did that research. Has there been some failed trials that scientific analysis was still considered helpful despite the drug not going to the FDA and being approved?
Sure. When and we actually used one of those. But the fact is, when we do the analysis, remember when I described analysis, there's a primary outcome and then there's secondary outcome. And I didn't talk about the exploratory outcomes. And we try and include all of those when we do a trial. So when we do the analysis, we look at the exploratory outcomes. And one of the exploratory outcomes is, does it affect the cell it's supposed to? And what about that genetic signature I talked about? So for abatacept, one of the exploratory outcomes was, did the genetic signature define who could respond? So we always end up looking at the pathogenesis of the disease, understanding what kind of cells do what under what circumstances. And that nearly always helps us understand what we're going to do next. And if we understand the disease enough, we may even get to the point of curing it.
Great. Thank you.
There were some other questions. I think
Yes.
Sue had some or something.
So there's one from, how do you pick which medication to start looking at in more depth? And how do you pick the study design? And also, are there any studies that focus on GI?
So that's a mere 3 or 4 hours of lecture, no problem. So I think one of the things is, remember, you've seen me and others talk about the pathogenesis of the disease. Those things that we think are important that cause the disease. It's drugs which affect different aspects of the pathogenesis that we choose to use. Because drugs affect all sorts of things, not necessarily in the pathogenesis of the disease. So we choose drugs based on understanding what causes the disease, what the pathogenesis is, okay? The trial design depends on what the drug does. For example, if we came across another prednisone, okay, which works really quickly, a couple of days, a week, well, our trials don't have to be 6 months. They could be 2 weeks long. On the other hand, if we came across a drug which affected a cell which works slowly, then the trial has to be very long to allow that slow acting drug to affect the cells that affect the cells that affect the cells that finally have a clinical response. So design depends on what the drug is all about.
Okay, thank you. She also mentioned, are there any current GI studies that focus on, studies focusing on GI?
Yeah, GI tract's been really very resistant because its pathogenesis is not very well understood. But there are definitely trials being done. And, you know, there was a wonderful abstract this year. I don't know if it was at EULAR or ACR, but it looked at a drug which is used for the lungs, but because of the way it works, may work for the GI tract. And so we're going to be doing that. And then, remember IVIG? Yeah. Very expensive drug. Never could figure out how it works. Give you a feel for it, it's about $250,000 a year. But it has been tried in scleroderma, and it looks as if it too may affect the GI tract. In fact, we're going to start a trial of it, but because of COVID, we've had to stop for now.
Thank you. Next question. Should all diffuse patients have a skin score? Do you have to go off all meds to be in a study? We kind of touched on that, but you can answer it.
Yes. So first of all, my belief is that every patient should have a skin score every time they come in, or at least every second time. I think it's a critical part. Why? Because the way the skin is involved affects how people feel, how they feel about themselves, their self-image, their ability to function. the pain with which they function. And if we know the skin score, we know what we have to do to try and make it better. So I believe very strongly in doing a skin score, even though it is not the primary outcome in most trials anymore. Okay? And we talked about the other part.
Yes. So there's two I'm going to combine about COVID. One sent to me and one online here. Is the COVID vaccination indicated for patients on immune compromised medication, and they have CellCept? But another one kind of similar is I'm wondering what your take is on the upcoming COVID vaccine for scleroderma patients. Should we wait to receive it to ensure that any potential side effects will not be harmful to us because we're immunosuppressed?
Okay. So this is another 30-minute.
I know.
But bottom line, you know, there were 75 different vaccines being tested. We're now down to about 9 that are really very much likely to be good, and 5 that are in higher dose in phase 3. And as long as the vaccine is not a live vaccine, okay, it is appropriate, just like the shingles or the flu. Is it going to work? Well, the first couple of trials look very hopeful, don't they? With 90% responses-ish, okay? How long that response lasts, we don't know. We're probably going to be using, in fact, we are going to be using 2 doses. It has a lot of downstream effects, but whatever, the need for it in patients with scleroderma is very high. If you give the vaccine, is it going to flare the disease? As far as we can tell, no. Is it going to give protection? Well, it's going to give a little less protection than people who are without any illness at all, about 10 or 15% less. But still, that's darn good. How long is it going to last? Well, probably as long as in normals, although the reason it doesn't work quite as frequently is because there's immunosuppression by the drugs or the disease. And so the body doesn't respond completely normally. And so we don't know exactly how long, but the expectation is it'll last just like flu. You get a dose every year or a year and a half, and I think it'll do just fine. Do I believe it ought to be used in scleroderma patients? The answer is yes, and early.
Good. Thank you. Straight and to the point. Last 2 questions. I notice trials usually only recruit patients with only a few years of diagnosis. This is a good question. What about those who have had scleroderma for an extended period of time?
So remember the first trial we talked about with collagen? That worked in patients with later disease. I explained why we used early disease in order to keep the trials relatively short. But ultimately, we need to have trials in later disease. And drugs like collagen type 1 may be the perfect drug for that. It's a little harder to do, but we definitely will be doing it.
So are you saying that perhaps in the future they'll have more trials that might give people who have had scleroderma longer an opportunity to participate in a clinical trial?
Absolutely. That is my definite hope.
Good. Thank you. Are there any promising Raynaud studies? Any tips for getting into a study?
There are, of course, a whole bunch of Raynaud's. The one that's going to be starting up very quickly is the one on iloprost. It's a drug that's been approved in Europe. It's being tried actively. There are a number of centers. Tina, do you know the company? I don't know the company, but the drug is iloprost. Oh, yes. The company is ICOS, E-I-C-O-S. I'm sure that if you look it up, they'll tell you who in your general neighborhood is doing it. I'm involved in the, I'm running the steering committee and so on, so I will not be entering patients, because that would be a ethical violation. I can't be doing both. But Dr. Kafaja is doing it. And so I'm sure that she would be looking for patients for that.
Thank you. And most likely, a lot of these are found on the clinicaltrial.gov page, right?
Good point. They're all trials.
Okay. And I can send that when I do my recap to everybody so you could take a look. And anytime we get that information, we'll share it as well with our support group leaders. And that was
And the other thing is, we're trying to arrange a way so that the Sclera Sun can inform people of ongoing trials. I don't know at the stage at which that is right now, but it is something we are considering.
Yes, we did send it out in our, not this newsletter, I think the one right before it. And we got a hold of a lot of the investigators, try to get an idea of the open studies and where in California they were being, that they were still recruiting. And we'll continue to do that. I think it's a really important area that we should continue in our newsletter. So I appreciate you mentioning that.
I guess I better turn it off and let my wife
Yes.
on. Huh?
Okay, thank you very much, Dr. First. We really appreciate this presentation. Well instructed and something that was very beneficial. And now we are going to give the floor to Elaine First, and she's going to be interviewing Brenda and Lisa. Elaine, take it away.
Hello, and I'm so glad that you are here. And I'm extremely thankful to Brenda Dunham and Lisa Brennan for being here to answer questions, my questions, and maybe yours too, about what it's like to be a participant in a clinical study. So I want to tell you that the point of this is not only to inform you and to give you some really great stories, if they have them, and give you some information about this, but also to recruit you in case you've had some misgivings about joining a study. Maybe Lisa and Brenda can help you with your misgivings, because scleroderma being an uncommon disease, when there are studies to be done, it's a lot of times very difficult to find enough patients to make the study a viable one. So every so often we do this kind of thing and try and get folks interested in joining studies when they happen. So let me start with Brenda. Well, let me just ask you a question. How many trials have you been in?
I've only been in one.
Okay, so it was this one.
Yes.
Was it the lanabasum one?
Yes, it was.
Okay, cool. And why did you volunteer?
Because I trust Dr. First immensely. I had gone through other doctors and no one was able to diagnose me or help me out. So when I found Dr. First, he was very open and honest with me, and I felt very safe. I was comfortable. I would ask him, would you put your kids through this? What would you do, in a child situation? And he reassured me that he would let his children do it. And I guess it's the rapport you have with your doctor and the sense of security that knowing he's looking out for your best interest.
Okay, so you did it because Dr. First said, would you be interested? And because he's a good doc and you trust him, then that was okay with you.
Yes.
So so that's how you chose the study that you volunteered for?
Well, I was fairly new to the disease. I think I had it for about 3 years before I discovered Dr. First, and he really sat me down and said it before. It was just kind of, oh, you just have Raynaud's, you just have this, you have that. Because I had 2 different doctors in Las Vegas. So when I found him and he sat me down and with me, I was assured that in fact I was with the right person and I had what he was telling me that I had.
Okay. And so when you called them or signed up online, what was the process to get you in to the study?
2016 that I started it, but I think it was fairly simple with Omar and Dr. First. I think it was just maybe going online and putting in my information, but the paperwork that was in the office, they went over it with me and then I just signed it. So it was something very simple to do.
Okay. And what did you like about being in the study?
I enjoyed being in the study because I want to find a cure for scleroderma. I felt like it. To Tina yesterday, was never really into the Scleroderma Foundation and what it was actually doing and the drugs. I found on Facebook that I didn't know about the CellCept was helping in one way, and then the CBD one was helping in another. And I enjoyed doing it because I felt like it was helping. I was seeing a difference in my skin. I used to be very, very shiny and very, very stiff. And now my face is just, like a normal, besides the tightness, I didn't have that shininess anymore. So when I started, I didn't realize the difference of what was going on and what was helping me and what wasn't. I did feel like the CB drug did help me. And when they told me that they were not going to approve it and they were cutting it off, I became very nervous because I'm like, oh my gosh, I feel like it's been helping me. So please don't take it off and let me go back, revert to what I was in the very beginning. It made me a little nervous.
And what's happened? Did you revert back to the very beginning?
No.
No?
I've stayed just the same.
Mm-hmm. Well, and so you're saying that your volunteering made you feel as though you were doing something important for the future of
Yes.
others.
Right.
Nice. I guess one of the things you didn't like about the study was having to go off it.
No.
If you felt that it was doing good things for you.
I was
Scared.
a little different. Yeah, scared. But when I started taking it, we just kind of went off of it this year. I was feeling good. I was feeling different than I was when I first came in being diagnosed.
Yeah. So what didn't you like besides that of being in the study?
I didn't really have a dislike of anything. I know when I was first on it, it was the blind study, the placebo or the drug. And I knew right off the bat that I was on it because I could feel some of the side effects. I'm not a partier in my age, but back in my youth, I know, kind of what it feels like to be foggy. And I started feeling that. So we went down from, or you call me a lightweight, but I went down from 2 meds from the morning and night just to the night, just so that I wasn't having those kind of side effects. So I really didn't dislike it. I enjoyed taking it because I felt like it was helping me.
Yeah. Yeah. Well, would you volunteer again for another study?
Of course. I told Dr. First I'll go to Italy with him anytime he needs someone. I'm like, look, it's my time. Let's go. We'll find a cure.
Cool. And so is there anything you want to say to any of the potential volunteers?
If you have the ability to do it and someone feels strong enough to help you do it, I would jump into it. Because a lot of patients I see on some of my social media pages or on the scleroderma pages where, one doctor's saying one thing and everyone's going, reach out for a second opinion. And that's what you need to do. You're your best advocate because you know your body. And after going through 2 or 3 doctors in Vegas, when I found Dr. First, he's my go-to. So it's finding someone that you trust and knowing that you're going to get better because they care about putting you into that study. They know what the outcome would be. Like out of the 3 trials, I was put into the CB one. So for some reason, Dr. First felt that was the best fit for me.
Mm-hmm. Cool. Well, there's a question about how many times you needed to go in to get to be part of the study. How many times did you go in all together for the study?
Well, I can't tell you how many. It was like
I don't want to be on camera, do you?
every other month I was in, every 2 months,
Yeah.
because you had to go do blood work. Dr. First always did my skin score. And then you had to do a urinalysis, and then you had to do the lung test.
So it was pretty often.
It was, yeah. And it
And
was,
do you, go ahead.
I was going to say it was a problem for me. I'm still employed and my employer's very understanding. So just the distance from where I live and Dr. First works, it's about an hour drive. So I'm in the San Fernando Valley and he's down in LA. So they were very understanding with my situation and my appointment. So we just scheduled it like on the Monday. So we knew which Monday I was going in, and I would just go into work late if I needed to.
Cool.
Yeah.
So what did the frequency of that bother you?
Only the 405 drive at 6 a.m. It was crazy. Like Dr. First, really 6 a.m. But that was it. But other than that, it was fine. I enjoy going to the office to see Omar and Dr. First. I love them.
Good. Okay. Is there anything else you want to tell people about joining a study?
No, I would just say do it. If you can do it, it's covered. They pay for it. I didn't have to, go out with co-pays when I had to go have blood work done or my pulmonary function test. It's all covered under the study. Plus the medication they give you is covered under the study. And I'm not quite sure about other ones, but they kind of give you a kickback, a little bit of money to do it. But I mean, that's not why I did it. But if anything that's going to help you, that you don't have that, that it is, and we don't have to have it approved by your insurance, I would definitely do it. I'm waiting for the next one. He said something else is coming up about put me in it. I'm in it. Let's go.
You're going to be a professional study patient.
A study patient.
Oh, cool. Okay. Do you continue seeing him? I mean, as part of the study?
Well, I'm out of the study now. I started seeing him at UCLA before he left for his private practice. So I'm ongoing patient. As long as he'll have me, I'm still going.
Okay, yeah. Cool. Okay, thank you very much. That was terrific.
And yes,
appreciate it.
I will always be glad to see you, by the way.
Thank you, Dr. First.
Okay, now Brenda, where are you? I can't see you on here.
Oh, Lisa?
Lisa.
I'm sorry, Lisa. Yes. How are you doing?
I'm well. How are you?
Good. I'm glad to see you, and I appreciate your taking the time to do this.
No problem.
So you heard the questions,
Yes.
right?
Yes.
And how many trials have you been in?
I've only been in the one.
This one, the
Mhm.
same one?
No,
Lanabasum?
for Ephedroban.
For what?
Ephedroban.
Ephedroban. Okay. I don't know what that drug is, but so why did you volunteer?
I think just since I just wanted some hope at the time, like I was not even a year diagnosed, and I was experiencing just like tendon friction rubs and just pain and muscle weakness and just like a whole host of things. And I was like, this can't be it for me. And so just, I was wanting to feel better, hopefully, find a better treatment, just hoping for something to improve my symptoms.
Okay. How did you choose the study you volunteered for?
So I had two presented to me, and one I believe was a safety trial. So, they were still trying to figure out dosing and a lot of other things. And then this one, with that particular trial, it was more visits coming in. It was also going to be administered via IV. And I was what they considered a hard stick. And I was like, multiple visits and I'm a hard stick. And I was just like, that doesn't seem like the right trial for me. Like I'm working and I'm like, no, I'm okay with that. And this other one was presented to me and it was mostly just taking pills from home. Like I had to go in for several visits. So I think it was a total of 7 visits. So the screening and then baseline and then the rest of the visits and a follow-up. So I think it was a total of 7. But like I said, for the most part, I could just, take the medication at home. And I was like, that will work. That will work. And then even with having to go to the visits, my employer was also really, really supportive of me. And she was like, just let me know as far as you can in advance when you need to go in for the visits and we'll, reschedule your clients and whatnot. So that was really helpful. So I think it was just more about convenience and like what they were actually studying. And so it was like, that works for me.
It's a good fit.
Yeah.
So it sounds as though you asked a lot of good questions to
I did.
find out what would fit for you.
Yeah, I prayed about it. I read through all of the material that they gave me. I talked to my mom and, just try to get as many opinions, not as many, but just opinions that I trusted. And then Dr. Kafaja was really helpful with answering any questions that I had as well. So that was
Cool. So how did you find out about getting this into the study? Or did she ask you?
She asked me, yes.
Oh, okay.
Yeah.
Okay, cool. And sounds like you have trust with her as well, like Brenda was saying.
Good for her.
Yeah, I don't blame you. She's a lovely person. Okay, what did you like about being in this study?
So a couple of things that I liked about the study were, like I said, at the time I was feeling not so great, and I felt like I was feeling better as I was taking it. Like it wasn't like a huge difference, but it was better than what I had been feeling. I like that, everything was covered. So I just had to get there. Yeah, I think those were the biggest things. Like everything was covered. I had a good relationship with Dr. Faja and the coordinator. They're very helpful. They're like, these are our numbers. If anything comes up, feel free to reach out if you have any questions. If you notice anything that's starting to bother you, let us know. So they were just like really upfront and supportive. And then, like Brenda said, this wasn't my main reason why, but there was like, you got some reimbursement. And I was like, I don't necessarily care about that, but I'm like, it doesn't hurt. So
Yeah.
I think those were the biggest reasons why I chose what I enjoyed about the study.
The reimbursement was for like gas money?
Yeah, it was like, I think up to like $75 per completed visit for parking and gas money, I think.
Yeah, and a couple of snacks.
Yes.
Get through there. Okay. What didn't you like? What was hard for you?
So I think this is just part of maybe having scleroderma, but so there were cardiac MRIs and I did not like that. These were my first MRIs in general, but in my head, when you go to MRI, most people are just like lying there and not really participating. And this, you had to participate. So you're holding your breath for any given number of seconds, and like you really had to be focused in there. And I think just the loudness of the sounds and just like, and I have to participate, it was just a lot. And then the other thing is just, I don't feel like I got a lot of feedback back about, what happened with the study or where it is, even now after I finished it. So I think those were the main things that weren't particularly enjoyable. And then just sometimes when I was there, just the amount of time that I spent at the hospital, which was, I guess, beneficial in a way because I was able to get most things done in one day, but it was just a long day, a lot of days. So
yeah. Okay, so it was the type of study that it was.
Mhm.
Well, you've got experience now with MRIs, eh?
So it was like MRIs, PFTs, skin biopsy, echocardiogram, 6-minute walk test, a couple other things I'm probably missing, but those were like the main things that I actually had physically to get tested for. So
it wasn't just taking pills.
no. So
Yeah.
like I said, but it was like within those 7 visits. So it's not like I was doing those all the time and they weren't every visit. Some were like the 2nd, 4th, and 6th visit or something like that. So it wasn't like every visit, all these things were happening, but
Okay, cool. Okay. Would you volunteer again for another study?
Yes. Yes.
Why?
Like I said, if I felt like I said, when I looked at the two different studies, if I felt like one fit into my life a little bit better, and I felt like looking at it, I was like, okay, I can do this, then for sure. But I'm like, in general, I would not be opposed to doing it again. I would definitely be open to participating in another study for sure.
Well, it's different now because you are not hurting as much.
Yes.
So that one of the reasons wouldn't be the reason now, right? But so
Sure.
now you've gotten your altruism up. You've got inoculated with more altruism that you probably already had, but it's now a little more to the forefront. Cool.
Yeah.
Cool. Anything you want to say to any potential volunteers?
Like I said, just consider it for sure. If for nothing else, like, especially if you have a trusted doctor and they're presenting this to you, definitely consider it. Or even if you're just like looking through some of the trials, like just consider it, like for yourself as well as for others. Like it can change your life. It can be a great help to you and those around you. So I would say just, like I said, consider it.
That's so sweet. I love that. Is there anything else you'd like to say?
No, I think that's it.
Well, it was a pleasure meeting both of you, and I appreciate your volunteering to be subjects today. I think you've probably done some really good educating today, and we appreciate that. Good to meet you.
Likewise.
You too.
Thank you, Lisa. Thank you, Brenda.
You're welcome. Thank you.
Very welcome.
I'm going to say a couple things and then I'm going to hand over to Dr. First so he can close out and say something before we end the meeting. And what I wanted to say is that in my experience as a research coordinator and listening to the two ladies talk about their experience participating, there were things that I noticed that I thought I would bring up, and I'm sure that the two ladies can attest to it, which is this really great relationship with your doctor, a great relationship with the team, your coordinator, the easy access in reaching your doctor, sort of you're in a more of a private forum with them where you don't have to go through some of the rigorous attempts to get a hold of your physicians through like these portals. And I think that being on a clinical trial gave a lot of people an opportunity to learn a lot about their disease. It helped them kind of be their own advocate and understand why these tests are important, what they're doing, and gave you a sense of purpose. And at the end of the day, everybody that participates in clinical trials, you help others that can go to a pharmacy today and pick up a drug. Because of you and the things that you've done on a clinical trial, we have these things being approved by the FDA, and you're picking them up at a pharmacy. And I'm seeing, and Dr. First and Elaine and everyone else, people live long, productive lives with scleroderma because of the leaps and bounds that have been made in clinical research.
Again, I just wanted everyone to hear all the lovely things that can be done through clinical trials. I think one of the things that I am always very impressed with is that the patients in the clinical trials are not doing it just for themselves. They truly want to help others. I hear it all the time. And it just says how really great you all are to try and help not only yourself and everyone else, but everyone else with scleroderma. So I thank you all for that. And I wanted to be sure you heard it.
I appreciate that, Dr. First. Thank you.
Take
All right,
care.
Dr. First, looks like we've answered all the questions. I appreciate your time today, Elaine. I thank you very much for your time today.